Good morning, all. I hope you're doing well this morning, and thanks for joining in. This is going to be our next to last lecture, and it's going to be about COVID-19 today. I'll tell you about the virus, I'll tell you about the vaccines, I'll tell you about the epidemiology of the virus and treatment, and so on. So on Friday's lecture will be on biotechnology, and one message that I wanted to convey to you, because I got a lot of email about this, is about the extra credit. So extra credit assignments can only benefit your grade. They cannot make it worse. Unfortunately, web courses does not accommodate treating extra credit as extra credit. So if you did not submit it, you will get a 0 for it, and you will see that your grade is affected. However, I will not rely on web courses to calculate the final grade. Okay, I will export your grades from web courses to a spreadsheet, calculate the real final grade according implementing the rules outlined in the syllabus, and treating extra credit as that, as opposed to an assignment which can worsen your grade. And then re-upload your grades with a new column, which will be called the real final grade. Okay, so do not look at web courses. Do not attribute you can see your grade on web courses, but keep in mind it's going to be a bit lower than your real final grade. If you want to calculate your real final grade, please look at the syllabus and implement the rules in the syllabus when calculating it. All right, any questions so far? Okay, let's switch to the lecture. So the topic of today's lecture is COVID-19. And COVID is an acronym, meaning it's a shorter version of something else. And the something else is Coronavirus Disease 2019. So COVID is caused by the virus called SARS-CoV-2, meaning SARS stands for Severe Acute Respiratory Syndrome, and CoV stands for coronavirus, and 2, I don't know what's that about. So these viruses look like this, and they are spherical. They have a spherical shape with some proteins embedded in the envelope. And inside the virus, as you may remember from the previous lecture, there's some genetic material. There is RNA genetic material. So there are 3 kinds of proteins that are embedded in the viral envelope. And the first protein is E protein, and this E stands for envelope protein, and it's responsible for production and maturation of the virus. The second kind of protein is M protein, and M stands for membrane. And these proteins determine the shape of the virus envelope. And finally, the very famous S proteins, which are called, S stands for spike, and the spike protein give the virus its name. So because those spike proteins form a crown around the virus, and that's why it's called coronavirus, which corona is, I believe, Latin for crown. These S proteins, they are so unique they are unique to the virus, and to the coronavirus, that is. And the vaccines that are available today, they are aimed at synthesizing this S protein for the body to recognize it and to kill it, to eliminate it. And this way, the body builds immunity to protect us against the coronavirus in the future. But I will tell about it in more detail in a bit. All right, so virus transmission. SARS-CoV-2 can be transmitted from animal to human, this is a direct transmission, and from human to human. It's also a direct transmission. In fact, the first time the virus kind of appeared in humans, it was transmitted from the animals to human. We don't know for sure which animal it was. It's widely hypothesized it was a bat, but we simply don't know. There is no definitive conclusion which animal was it. Uh, there was some talk about pangolins. There were some talk about bats, but there is lots of studies going on right now to determine what exactly was it. And human to human transmission is primarily through infectious droplets that may travel up to 6 feet from person to person. And we emit these droplets through coughing, sneezing, or even talking and breathing. I mean, when we breathe, it's not always clean breath. Sometimes we emit droplets. However, with the new variants emerging, particularly the UK variant, it was found to be more contagious. And the reason it's more contagious is it's because it can be aerosolized and it can travel. It can travel for farther, and it can stay in the air for longer. So. The aerosolized particles, they can remain viable for up to 3 hours, so, which means like if you're going to the gym and everybody's kind of on the treadmill running, and if the windows are closed, that means that over time the air will contain more and more and more coronavirus aerosolized coronavirus particles, okay, or coronaviruses. So that's why staying in closed, in unventilated rooms is a bad idea right now unless you're vaccinated and everybody around you is vaccinated. All right. This is going to be very text heavy lecture, okay? So bear with me here, but it's on one hand, you may feel like I'm reading, which I will be reading, but on the other hand, you will have lots of text to refer to later on because I will post this in web courses. So virus transmission, indirect transmission can also take place, meaning not directly from human to human, like particle does not have to travel inside your mouth or inside your nose or inside your eye to get infected. Somebody can sneeze on some surface. Somebody can sneeze on a, I don't know, doorknob, or kind of sneeze on the hand and then touch the doorknob, and the virus will stay there with whatever droplet it was emitted with. And it can stay on the surfaces for long times. Like it was shown that on surface such as plastic, the virus can survive for 3 days. So even though somebody may have touched the doorknob yesterday, today you can still touch the same doorknob and get the virus. And if you don't wash your hands, and touch your face, then you can catch it that way. However, in the past couple of days, I think there was a news that the probability of indirect transmission or transmission from just the doorknobs or like groceries, it's pretty low compared to the direct transmission from person to person. Okay, so compared to aerosols and droplets that are transmitted directly from person to person, catching it from the surface is it has lower probability. So interestingly, viral particles have been found in fecal samples or poop, and there is accumulating evidence for fecal oral transmission. So, and this is the part where old wisdom and just a good tone rule of washing your hands after you're done with the bathroom is paying off, because if you wash your hands, then you will not transmit the virus to the door handles and other surfaces that you touch with the fecal particles on your hand if you don't wash them. Um, so the virus transmission is higher in the population dense areas, in large public gatherings and facilities such as prison, cruise ships, and senior living centers. Because they provide ideal conditions to spread the virus. People live in close quarters. They don't physically distance from each other, and it's often poorly ventilated areas. For cruise ships in particular, like everybody's on the ship, everybody's eating from the same dishes essentially, and everybody's using the same kitchen. So it's that's kind of one of the reasons why cruise ships were the first ones to experience high spread in the coronavirus disease. So, how does the virus enter the host? the host being human. So portals of entry include the respiratory tract, the eyes, and by respiratory tract I mean the nose or and the throat. The If we rub the eyes or somebody coughs or sneezes in our eyes, the droplets can travel to the eye and infect, and the virus can get into our system through the eyes and infect everything else. We can inhale the virus particles, and it can infect the oral. It can start. The infection can start in the oral cavity and esophagus. So once transmitted, viral S proteins bind to an enzyme on the host cell, and this specific enzyme is called angiotensin converting enzyme 2, or uh, the acronym for that is ACE2. Okay, let me check the chat here. Christine says, does chlorine help kill the virus? Yes, it does kill the virus, but please be careful with chlorine because it's poison. Okay, so definitely don't drink it. Unlike, there were some rumors and some ill advised methods of fighting the virus, like drinking chlorine. Do not drink chlorine, okay? And be very careful with what you put it on, because even if you decide to disinfect the groceries, which latest studies show that you may not need to do because the probability of virus transmission that way is low. Um, so even if you disinfect the groceries and decide to rub the apple with chlorine, you will then eat the apple. So don't do that. You will get poisoning from that. Um, okay, let me check the chat again. Ba, 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 ba. So a water park is not a good place to go. No, it's not, unless you're vaccinated and everybody else is, or rather most people are. CDC is kind of changing the guidelines right now. It's like a couple of weeks ago, they released new guidelines on social gatherings. And so it's now safe to gather if all of the people are vaccinated. So if all of the people are vaccinated, you're free to go, like to have parties again and socialize. But if people are not vaccinated, you still have to wear masks and socially distance. Because when you're vaccinated, even though your probability of going to the hospital is super low, you can still transmit the virus to unvaccinated people, and that is why it's important to still wear a mask once you're vaccinated. But I want to go back to the virus transmission. So the S protein, the spike protein on the surface of the virus, it binds with ACE2 enzyme. And this ACE2 enzyme has several functions in the body. It lowers the blood pressure, it controls fluid balance, and it regulates the inflammatory response. And it's present in a lot of tissues. This ACE2 enzyme is present in a lot of tissues, including lungs, kidneys, heart, arteries, and the gastrointestinal tract. So that is why once we get the virus.
1 second.
Yeah, go ahead. Oh, Ava, were you talking to me or somebody else?
So sorry, someone was at my door and I accidentally unmuted.
Okay. Because ACE2 enzyme is present in a lot of tissues, if a person is infected with the virus, the symptoms can appear in many organ systems at the same time, or gradually travel from one organ to an organ. So the person can feel the symptoms in the kidneys. Kidneys can become inflamed and cause a lot of trouble. The heart can be inflamed and that might even lead to because the ACE2 enzyme is also in arteries, it can lead to blood clots. And people often experience nausea and diarrhea if they have COVID, and that is because ACE2 enzymes are also present in the gastrointestinal tract, and virus can infect cells in the gastrointestinal tract. So once the virus binds to the ACE2 enzymes on the cell the cell either engulfs the virus, or it's the endocytosis of the virus happens, or the membranes of the envelope of the virus and the membrane of the cell fuse, and viral genome enters into a host cell. So mechanisms of pathogenicity. What happens next? Once inside the host cell, there are three stages of what's happening next. Stage one, the virus takes over the replicative machinery and multiplies within the cell. And think back to, sheesh, like 20 lectures ago, we kind of discussed this. We discussed what happens once the virus enters the cell. The virus genetic material kind of hijacks it piggybacks, right? It doesn't hijack, it piggybacks on the cell's own replicative machinery. Like messenger, remember the messenger RNA is synthesized, and then this messenger RNA goes to a ribosome, and ribosome produces the protein. So once virus enters the cell, its genetic material is replicated, and its genetic material, its genes are expressed. So virus needs to synthesize the S proteins, the M proteins, the E proteins. So all of those are synthesized within the cell, and then the proteins and newly replicated genetic material, they assemble into the new viruses, and and then viruses ruin the cell and exit the cell and infect the neighboring cells. So viral copies or progeny, that's the final stage, they are released from cells through exocytosis and infect the nearby cell. So stage two. Stage two deals with the immune system response. Whenever the cells that belong to the immune system that circulate our body, whenever they detect the foreign protein in our body, they panic and they trigger the inflammatory response, calling help from another immune cells. Okay, so immune system triggers inflammatory response. If not blocked by the immune system, and this immune system response, inflammatory response, is aimed at killing the cells that are infected with the virus. Uh, well, that's what happens usually. So, if not blocked by the immune system, the infective form of a virus outside of a host cell, they migrate to the lower respiratory tract where they infect the alveoli cells. And does anybody remember what are alveoli?
They're the smallest part of your lung that's filled with air or whatever. I forget like a formal definition.
So alveoli are located in the lungs. Okay, so um, we have bronchioli, and at the end of the bronchioli we have alveoli. So alveoli are air sacs that are that have lots of capillaries, and that is alveoli are the site of gas exchange. So within the alveoli, CO2 leaves the capillaries, and oxygen enters the capillaries. Okay, so these are very important little structures for us to get oxygen and get rid of CO2. So the alveoli are very rich in ACE2 proteins, and that is why virus readily binds to those cells and eventually ruins them, interfering with our breathing and our well-being. So extensive exocytosis of virus particles leads to apoptosis or programmed cell death of alveolar cells. And if we have less and less and less alveoli, I'll say that, um, it's it impairs our breathing. Alveoli do not stay like we our body regenerates alveoli. However, if the rate of death of alveolar cells is higher than the rate of regeneration, which is often the case with COVID disease, we are in trouble. Like our oxygen saturation goes down, and we have trouble delivering oxygen to the cells within our bodies. So the last stage of the COVID infection is the non-specific immune response. So the immune cells release chemokines, a chemical attractants to call white blood cells to the sites of the infection. So chemokines, they cause inflammation. Okay, and they stimulate the targeted attacks on the infective cells. And by inducing inflammation, this process basically cripples the normal cell function, because the inflammation, the target, like the immune system, it kills off the cells infected with the virus. So in critical cases, what happens when too many cells are infected with the virus, a cytokine storm can happen, and it's basically an overreaction by the immune system. It stops being specific, like the immune system stops attacking the cells that are infected with the virus. It attacks everything. And whenever immune system attacks everything at once, it's called cytokine storm. So in this way, the immune cells, they kill off cells that are in multiple organs within our body. And usually people who are in critical condition after COVID or during COVID, they are in critical condition because of the cytokine storm. It's often is a cause of death, unfortunately. So, what are the disease manifestation? There are also three stages of the disease manifestation, and not every stage, not everybody will progress to the second stage, and not everybody will progress to the third stage. So, stage one is an asymptomatic state. We carry the disease, or people carry the disease, but but experience no symptoms. The virus has some incubatory period, and its median incubation time is 4 to 5 days, but it can last to up to 2 weeks. And that is why whenever you enter to another country right now, like if I were to go to Canada, I would need to stay put in some place. Like I would need to rent an apartment and sit there and not leave the apartment for 2 weeks. And that has to do with the incubation time of the virus. So they want to make sure that I did not bring COVID with me. And if after 14 days there are no symptoms, then I'm free to go and socialize. So 14 days is the maximum incubation time for the virus, or the maximum amount of time that you may not exhibit symptoms after catching coronavirus. So at this stage, at the incubation stage or asymptotic stage, virus can be detected by a nasal swab and is shed by the host in the respiratory droplets. So even though you're not exhibiting symptoms, you're still contagious. And this is another reason why you have to stay quarantined for 2 weeks if you travel somewhere. And it's kind of a good idea if you travel from state to state to also quarantine. It's not technically a requirement, but if you care about the people around you, that's a good idea to do that. And so progeny are released and propagate or spread, and there's a mild innate immune response initiated. So at the end of the asymptotic state, well, as if we don't feel the symptoms of a disease, it doesn't mean we're sick. If we have a very mild immune response, we may not even have a fever. We may not even feel it, but we can have like mild inflammation inside our bodies that is killing off the cells that are infected with the virus. Now, if immune system did not kill off all the cells with this mild innate immune response at stage one, the disease progresses to stage two. So during stage two, people start experiencing upper airway and conducting airway response. So the immune response becomes more robust, meaning the fever appears, and you start coughing, you start experiencing shortness of breath, chills, muscle pain, loss of taste and or smell, sore throat, nausea, and diarrhea. So this is stage two. So immune system did not kind of do, could not complete the killing off of the virus in stage one, and in stage two, it ups, it kind of enters into a higher gear, the immune system does, and you start like experiencing fever and chills, and the rest, cough, shortness of breath, muscle pain, loss of taste and smell, is the effect of the virus itself and the cells dying. So up to, the good news is up to 80% of COVID-19 cases will be either at stage 1 or stage 2 and will not progress to stage 3. However, 20% of cases will enter stage 3, which is serious business. And during stage 3, people experience hypoxia or lack of oxygen saturation of the blood, and they may progress to acute respiratory distress syndrome or ARDS. So, and it happens within 8 to 12 days. And at this stage, at stage 3, critical care and hospitalization is necessary. And approximately 15% of COVID-19 patients require oxygen because the cells in the alveoli are dying off faster than they're regenerating, and which means that the gas exchange is impaired and they need help oxygenating their blood. And 5% require ventilation. So manifestation at stage 3 include pneumonia, difficulty breathing, persistent chest pain or pressure, confusion because of the lack of oxygen, inability to stay awake also because of the lack of oxygen, bluish lips or skin also because of the lack of oxygen. Oxygen levels fall as lungs become filled with fluid and white blood cells, mucus, and cellular debris. And if the person experiences cytokine storm, it will lead to dramatic drop in blood pressure, leaky blood vessels, formation of blood clots, and organ failure. So cytokine storm, again, is an overreaction of the immune system to the virus. Like there's so much signal going to immune system to protect against the virus that immune system is like, okay, well, I'm going to kill everything. And it does. So what are other disease manifestations? The virus virtually affects all of the systems in our body. It doesn't only affect the lungs. As I mentioned before, the virus binds to ACE2 proteins on the cells, and ACE2 proteins are plentiful across multiple organ systems in our bodies. So if a person has COVID, they can manifest. It can manifest as conjunctivitis or pink eye. On the nose side, patients lose their sense of smell from viral damage of nerve cells in the nasal passage. Pneumonia is a common manifestation at stage 3. So liver is also affected. The overactive immune system and therapeutic drugs affect the liver enzymes. Kidneys are affected. Remember, kidneys have, the cells in the kidneys have ACE2 enzymes, which are the target for the spike protein. And damage is common in severe cases, either from direct viral attack or multi-system organ failure from the cytokine storm. Cardiovascular system is affected. Virus directly infects the cells that have ACE2 receptors, leading to clots, inflammation, heart attacks. Some patients experience confusion, seizures, inflammation, and strokes. So the virus affects the brain. And as the time progresses and the patients are studied, another symptoms of the disease is interestingly depression. So people who have recovered from COVID, one of the frequent symptom of long COVID, so quote unquote, is depression. In the intestines, GI tract is rich with gastrointestinal. GI stands for gastrointestinal tract, is rich with ACE2 receptors, and 20% of patients experience diarrhea. So long COVID is a term used for disease manifestations that linger long after SARS-CoV-2 has been eliminated from the body. So even though there is no more virus in the body, the body still is recovering. And I don't remember the percentage of people. It's not very high. Like I remember that 40% of people experience long COVID in terms of fatigue. Like it takes a long time to get back to normal. People usually are fatigued after experiencing COVID, but I don't remember like the percentages for other long COVID symptoms are lower than that. So a lot of people still experience cough and shortness of breath, even though there's no more virus in their body. They experience headache, heart palpitations, chest pain, joint pain, physical limitation, depression, and insomnia. And it's not really clear why it's happening. And there's quite a bit of research going on now, like what's what happened, like what's the virus is not there anymore, and the immune system has no reason to overreact anymore. So why are people experiencing these long-term systems? So at this point, it's like a mystery, but hopefully more researches will happen soon, and we'll know what to do with this. So clinical care for people with COVID includes increasing oxygen in a less invasive way than a ventilator. People have found that the ventilator, although helpful, it's the outcome for patients who have gone on ventilator treatment was not great. The outcome for patients who were on more non-invasive treatment for oxygen supplementation, it was better. So doctors have moved away from the ventilator, and they're using it in extremely bad cases. Another another clinical care way is basically proning, or placing patient face down to expand the dorsal lung regions. People with COVID who have pneumonia, if they lay on their back, they have a hard time breathing. So just changing the physical position, lying face down instead of face up, or on your side, but or on your side, usually face down. So lying face down like changes your the shape of your lungs a little bit, and it helps breathing more easily. So another item for clinical care is prescribing drugs. And one of the drugs is remdesivir, which helps fight viruses. And another drug is called dexamethasone, which is a steroid, meaning it's anti-inflammatory drug. It reduces the inflammation. And prescribing dexamethasone, it reduces the chances of the cytokine storm, or overreaction of the immune system, where immune system kills everything in its sight. And another treatment is delivering convalescent plasma, where convalescent means the uh, plasma coming from people who have recovered from COVID. So people who have recovered from COVID, they developed antibodies. And if these antibodies are transferred to the person who is sick with COVID now and is in bad shape, then it's kind of a supplement. It helps the person's immune system to fight the disease by getting additional, like, quote unquote, troops from another person's system, and these troops being antibodies, the proteins antibodies. Okay, so the risk factors for COVID-19 are, so most of people, 80% of cases will not be serious. However, some, well, I'm saying it will not be serious, but there's long COVID, of course. So even though you may be not in a risk group, you may experience some unforeseen circumstances after getting COVID, in terms of like fatigue, shortness of breath, depression, insomnia, and so on. So not fun. But there are particular population groups that have higher risk of dying from COVID, or higher risk of multiple organ failure from COVID. And these population groups include individual aged 65 and older, especially those in a care facility where they live in higher density and in close quarters. Anyone with underlying medical conditions such as diabetes, asthma, kidney disease, pulmonary disease, blood disorders, obesity, or metabolic syndrome, heart conditions, they also have higher chance of not making it from COVID, and higher chance of experiencing harsher side effects from COVID. Also, immunocompromised individuals due to AIDS, cancer, blood or organ transplants, and immune deficiencies are also more susceptible to poorer outcomes from COVID. And this is because, well, their immune system is literally compromised. Their immune system cannot fight off the disease as efficiently as a healthy person. So we have, although a lot of people are healthy and may think, oh, I'm healthy enough and I can go out and get COVID and I'm not gonna, it's not gonna kill me. Well, it may not kill you, but it may kill other people who are around you and who are more susceptible. Like, you may see who is over sick. Some population groups are visible, like you may easily identify a person who is over 65, but a lot of population groups are in the, like, you cannot tell right away if the person has AIDS or has cancer or has blood or organ transplant or some other disease that makes them susceptible. So it's important to be to be socially responsible and in this time at least, and keep in mind that you cannot always see a person who will die of COVID if you transmit it to them. Okay, so it's important to wear a mask, it's important to get vaccinated, and it's important to kind of be in the know of how you can reduce the risk or minimize the risk of the transmission of COVID to other people. So another thing that I wanted to touch on is what was in the news back in April or May of the last year. And it was this basic reproductive number R naught. There was a lot of talk of R naughts and like how, what is COVID's R naught, what are other diseases R naught, and how can we bring R naught down? So first, R naught is an epidemiological metric that means that describes the number of individuals that can be infected from one single person. Okay, so if R naught is 2, that means that 2 people can be infected by one single person. If R naught is 0.5, it means that, well, half a person of average is going to be, like 2 people will infect 1 person. That's what it means. So R naught assumes that all members are susceptible to the infectious agent, and therefore R naught is not a constant number. It varies depending on the environment, like population density, and it varies depending on the behavior of the population. So reducing R naught, like wearing masks, is reducing R naught. Socially distancing is reducing R naught, and there was and still is a lot of talk about how we are reducing R naught, and it's basically saying that how can we reduce R naught below 1, because R naught of less than 1 suggests that a disease will ultimately die out in a population, because one person, if one person does not affect another person, the disease will not spread. So I'm not going to read all of that. I'll just say that R naught for diseases that spread via aerosols, like measles. Measles is extremely contagious. It doesn't spread with droplets. Like you can be in the same room as a person and be like 12 feet apart. But if you spend or I don't know, no, you know what, it's a bad example. You can pass a person with measles on the street and catch it. So this is how contagious measles is, and its R naught is 12 to 18, meaning one person can infect 12 to 18 people with measles. So Ebola is another. The R naught for Ebola is 2, and Ebola is transmitted directly through body fluids. So what was the R naught for COVID-19? So back in 2020, in May of 2020, the R naught for the original strain of COVID was 2.2 to 2.7. So it's not as contagious as measles, but a little more contagious than Ebola. So currently, we don't really know what R naught is for the new variants, and it's currently being calculated, particularly the more contagious UK variant, but it's higher than this based on the rate of spread. So R naught is crucial to determine what percentage of the population must be immune in order to halt the disease spread. Like with R naught of 5.7, at least 82% of the population must be immune, thereby preventing the spread through herd immunity. So at this point, it's estimated that at least 70% of people have to be immune to COVID-19 in order to stop the spread of the disease. And immunity is acquired either through vaccination or having had COVID-19. However, it should be noted that the immunity after you've had COVID-19 is not long-lived. Like you can catch the disease again after 4 to 5 months. After you've gotten better, 4 to 5 months later, you can catch the disease again. So the vaccines at this time, there is evidence that vaccines provide immunity for up to 12 months. So getting a vaccine is more effective at this point than having had COVID. So we can.
Excuse me.
Yeah.
I actually saw an interesting video on R naught about COVID.
Hmm.
They said it was hard to determine the R naught of COVID because the like certain super spreader events, like people would go out, and so like one person would affect would infect like 13 in one scenario versus like another person. Like the average would be 5, but then if you calculated R naught with 5, you wouldn't be correct because sporadically you have cases where it's 13 or 12 or what,
Yep,
etc.
yep, that's very true. And that goes back to the statement that R naught varies, and it depends on the population density. It depends on the environment. So if you've got the super spreader event, it's likely like the population density that affected the rise in the R naught. And if a person like lives in the rural area and gets out like once a week to buy groceries, R naught in that area is going to be lower because the opportunity to meet other people is low. Okay, so that goes back to the statement that R naught is it differs, like it differs based on population density. But good point, yeah. So if population density is high and R naught is 13, that is kind of evidence for the COVID being aerosolized and spread not with fluids, not with droplets, but through air, like measles, which is likely outcome with the new variants. So we can implement some personal safety rules to avoid the contracting and spreading COVID. And that includes washing hands often, using a hand sanitizer with over 60% of alcohol when washing is not an option, but washing hands with soap is more effective than using alcohol sanitizer. So we can maintain a safe distance from other outside of home of over 6 feet, and wearing masks. Wearing masks is important. And excuse me, CDC now recommends wearing 2 masks instead of one, with the more infectious variants out and becoming dominating in the US. And clean and disinfect frequently, touch surfaces often. However, this last point, in the light of the news in the past couple of days, it's not as effective. Like it doesn't do as much as wearing masks, keeping social distancing, and getting vaccinated. So before areas can safely open to the public, a disinfection plan must be implemented to prevent spread in areas that typically host high numbers like schools, public transit, aviation, shops, restaurants, and churches. Studies have shown that SARS-CoV-2 lasts hours to days on surfaces, less in the sunlight. So leaving items untouched over 24 hours or outside in the sun will basically disinfect the item. So there's on this website, there are EPA approved disinfectants that you could use. And the third one is UV radiation. I'm kind of speeding through that. You can read that later, but I want to get to vaccines. I want to talk about vaccines for a bit since we're 7 minutes away from the end of the class. So currently in the US, three vaccines are approved. And there are 3 types of vaccines that are out there or that are possible. So 3 types of vaccines that are possible for COVID are mRNA vaccines, viral vector vaccines, and protein subunit vaccines. Currently, there are no approved protein subunit vaccines, but there are approved mRNA vaccines and viral vector vaccines. And next, I'm going to explain a little bit how these 3 differ from each other and how these 3 work. So mRNA vaccines, does anybody remember what is mRNA? The final is soon and the questions will be there for mRNA. We've studied this. Aaron, messenger RNA. Yes, exactly right. And what does messenger RNA do?
It replicates the original DNA strand and carries it to the rRNA. Right?
It carries it to ribosome. Yes. So mRNA, it copies the gene for a particular protein, the instructions for a particular protein. It exits the nucleus and goes to a ribosome where this information this recipe for a particular protein is read by a ribosome and transcribed into the actual protein itself. So mRNA vaccines, they basically inject the messenger RNA with instructions to make the S protein, to make the spike protein that is, remember, the protein that is embedded into the envelope of the virus. And so mRNA vaccines, they, if you get an injection with an mRNA vaccines, you get an injection with the messenger RNA. So once the messenger RNA enters into your body, it carries the instructions for the S protein. Then your body starts making the S protein, but it doesn't do it for long because messenger RNA itself is attacked by the immune system and destroyed. Now, the protein itself, it's in your body and it's also attacked by the immune system. And immune system, it recognizes this, oh, holy cow, that is a foreign protein in our body. Let's get rid of it. The inflammation is triggered and the antibodies are formed and your immune system kind of kills off this protein. Now, once the protein is killed, the information about the antibodies, like this, the specific kind of antibodies that were used by your body to kill this S protein, it remains in your body. Okay, so your body kind of develops a memory how to delete, how to get rid of this S protein that comes with the coronavirus. So whenever the coronavirus enters your body, it doesn't take your body 2 weeks to learn how to fight it anymore. Your body can fight it off like within 3 to 5 days because there are memory cells in your immune system that remember how to fight this specific S protein. So there are currently 2 messenger RNA vaccines available. One of them is Pfizer, another one is Moderna. They're extremely effective for fighting the COVID, or rather deleting the S protein, like fighting the S protein. And they have like over 90% of effectiveness 2 weeks after the second dose. And this is important. So you're considered immune to the coronavirus if you were vaccinated with Pfizer and Moderna, or Moderna, 2 weeks after your second dose. Now, viral vector vaccines work a little bit differently. Viral vector vaccines use a harmless version of a different virus, which is called a vector. So this different virus is engineered to carry the instructions to make this S protein. So not the mRNA is injected, by a different virus with the edited genome is injected. And this edited genome of the virus is providing instructions how to make S protein. And the rest of the mechanism of response is similar. The body recognizes the foreign protein and starts attacking it and developing these memory cells in the immune system that that facilitate faster response to COVID infection later on. And there are 2 types of vector vaccines. One is Johnson & Johnson and another one is AstraZeneca. And only Johnson & Johnson right now is approved for use in the United States, and AstraZeneca is used in multiple other countries, mostly in Europe and I'm not going to say, like, Europe is the more prevalent area for the AstraZeneca vaccine that was developed in Oxford. And protein subunit vaccines, currently there are no vaccines that work well, but essentially the protein subunit vaccines, the protein itself is injected into the body, and the body is trained to find the specific protein just from the injection. But there's no vaccine that works so far. Johnson & Johnson was paused. Yes, Johnson & Johnson was paused. There will be a hearing on the 23rd about the evidence of the adverse effects associated with Johnson & Johnson vaccine. So what was the deal with the stopping of the Johnson & Johnson vaccine was that after administering 7 million doses of Johnson & Johnson, there were 6 cases of a rare disease, or rather not rare disease, rare immune reaction that was associated with low levels of thrombocytes and high levels of clots, which is kind of counter blood clots, which is counterintuitive. So the administration of Johnson and Johnson was stopped out of abundance of caution. But just to reiterate, 6 cases of this adverse reaction versus 7 million administered doses. Okay, so this the probability of this adverse reaction is super low. So at this point, it seems that the benefit from administering the Johnson and Johnson vaccine is higher than the cost. So one last bit that I wanted to mention about the viral vector vaccine, it's their effectiveness is lower compared to the mRNA vaccines. However, they only require 1 dose. It's like the effectiveness is like 66% after 2 weeks of 2 weeks after the vaccine administration, both for Johnson and Johnson and AstraZeneca. So yeah, I'm going to stop here because it's 10:21. Does anybody have any questions? Will we be able to review DNA and gene structure before the exam? I can include it in Monday's lecture. Yeah, but you will have the study guide for the final exam, similar to other exams, and you have the recorded lectures that are posted on in modules section on web courses, so you can review the slides and review the videos. And on Monday, I'm going to cover some of the topics. Any other questions? All right. Well, thanks for joining, guys, and I'll see you on Friday when I will talk about biotechnology.